Hello children, let us proceed our chapter, the fundamental unit of life. And today we are going to discuss about various cell organelles. So what are cell organelles? Let us recall that these are those small cellular compartments which are bound by their own plasma membrane. Some of them do not have their own plasma membrane, but the main thing is that cell organelles are there to perform definite functions, and that's how the division of labor within a cell exists. So due to this division of labor by various cell organelles, a cell becomes very efficient in its functioning. So the first cell organelle that we are going to discuss is endoplasmic reticulum. We will also emphasize which of these cell organelles are single membrane bound, which of them have got double covering. And so far we have only discussed one structure in the cell, that is nucleus, which has double membrane covering. So endoplasmic reticulum is a single membrane bound structure. Now how does it look? It looks like it has various tubes and sheets, and it is in continuation with the outer membrane of the nucleus. Then there are two types of endoplasmic reticulum: rough endoplasmic reticulum, on which you can find these dot-like structures, and smooth endoplasmic reticulum. The rough endoplasmic reticulum looks like this because these dot-like structures are ribosomes which are attached on its surface. Now children, ribosomes are those cell organelles which perform protein synthesis. So rough endoplasmic reticulum is involved in the synthesis of various kinds of proteins with the help of ribosomes. Then smooth endoplasmic reticulum, what does this one do? So it synthesizes various kinds of lipids. Now some of these proteins synthesized by RER and lipids synthesized by smooth endoplasmic reticulum together form the plasma membrane. If you remember, plasma membrane is made up of proteins and lipids. So this process is known as membrane biogenesis, and remember that endoplasmic reticulum is involved in this process. Also, some of the proteins synthesized by RER might function as enzymes. Some of those lipids might function as hormones and steroids. Also, smooth endoplasmic reticulum in the vertebrate liver cells have a specific function. They perform detoxification. So if by chance there are some toxic substances in the body of the organism of vertebrate kinds, then smooth endoplasmic reticulum performs the detoxification activity. So together, overall, endoplasmic reticulum is a network in the cell, and it channels various essential proteins and lipids to their designated targets, that is various parts of the cytoplasm. With this, let us move on to another organelle which is not only visible here but is often closely associated with the endoplasmic reticulum, and they form a part of cellular membrane systems. So this is Golgi apparatus. Yes, Golgi apparatus is made up of flattened sac-like structures which are known as cisterns, and often it is found very close to the endoplasmic reticulum. Let us learn why. Because its main functions are storage and modification and packaging and dispatch of various products. So some of the products that it obtains from endoplasmic reticulum through these vesicles will be either stored, or if they need to be dispatched somewhere, then they will be packaged into smaller vesicles and then sent to their target position. Also, they modify various compounds, like they can form complex sugars from the simple sugars. A very simple example is that Golgi apparatus is involved in the cell wall synthesis in plants, and cell wall in plants is made up of cellulose. Now cellulose is a complex carbohydrate. So the formation of this complex carbohydrate takes place in the Golgi apparatus. So you can imagine that storage, modification, packaging, dispatch and cell wall formation, these are the kind of activities which will be disturbed if the Golgi apparatus of a cell is not functioning. So this Golgi apparatus, finally we should also learn that is involved in formation of lysosomes. How? Because the enzymes which are present in a lysosome are synthesized in the rough endoplasmic reticulum. Enzymes are mostly proteins. So once those enzymes are synthesized here, they are transported with the help of a small transport vesicle. They come inside the Golgi apparatus where they are nicely modified or packaged, and then the small vesicles are pinched off from the cisterns which contain powerful hydrolytic enzymes, and once they are coming out in the form of a vesicle containing enzymes, that structure itself is a lysosome. So now we must try to understand that lysosomes are formed by the combined effort of endoplasmic reticulum and Golgi apparatus. With this, let us move on to lysosomes, because remember children, endoplasmic reticulum, Golgi apparatus and lysosomes are essentially very much related to each other. Now, lysosomes are known as suicide bags of the cell. We will learn in a moment why. But what are these? These are enzyme filled vesicles, as we have just discussed, and they help in the digestion. So remember, when the amoeba like unicellular organisms engulf some kind of food particle through endocytosis, that food vacuole is now fused with the lysosomes, and with the help of those enzymes the food is digested. Also, they help with the cleaning of the cell. So whichever cell organelles are aging, they are not functioning anymore, so they all are brought to the lysosomes where they will be digested so that the cell remains clean. Also, protection of cell from the foreign objects. So if there are bacteria and viruses entering the cell, they will be attacked by the lysosomal enzymes, and they will be destroyed to keep the cell healthy. Ultimately, they also function as the waste disposal system of the cell. So how do they do this? First, they can keep the cell clean by digesting all the unwanted or non-functioning organelles, and also they can break complex substances into simpler ones. So once they are breaking complex non-usable substances into simpler forms, those simpler forms can be recycled into making some other cellular structure. That's how they keep the cell clean and increase its efficiency. Now let us discuss that point where they are known as suicide bags of the cell. So if by any chance the cell has been damaged in such a manner that it can no longer function efficiently, then the lysosomes burst, releasing all their enzymes, and that's how they destroy the entire cell. And that's why this name has been given, because they destroy that entire structure in which they themselves are present. So with this, we are moving on to the next cell organelle, which is mitochondria. Very important cell organelle, because every process in this world, whether it is a living system or a non-living system, requires energy. So mitochondria is a powerhouse. It is a different matter that some cells which lack mitochondria also produce energy, but the amount of that energy which is produced is very little as compared to those cells which have this magical structure. So first thing to pay attention is that after nucleus, mitochondria are those structures, those cell organelles which have double membrane covering. So there will be an outer membrane and then there will be the inner membrane. Let us learn more about this. So remember, it is a double membrane bound organelle, and it also contains its own DNA and ribosomes. So there is certain level of autonomy in this organelle. Now, what is special about this? Why is this powerhouse of the cell? Because as I have told you that the outer membrane and inner membrane are there. The inner membrane is folded in the shape of finger-like structures. Now, these finger-like structures are known as cristae, and these cristae are folded structures, so the surface area of the inner membrane increases many-fold. And what do we do on this increased surface area? This is the site of ATP synthesis. So children, remember, cristae are the site of ATP synthesis, or inner mitochondrial membrane is the site of ATP synthesis. Now what is ATP? Its full form is adenosine triphosphate and it is the energy currency of the cell. Why? Because ATP are those kind of molecules in which energy can be stored once they are synthesized, and for whichever cellular process they are required, they will be utilized accordingly. So remember, ATP is also known as energy currency of the cell. And where is it synthesized? It is synthesized in the inner membrane of the mitochondria. And since mitochondria is that cell organelle which synthesizes the energy currency, that is why it is known as powerhouse of the cell. With this, let us come to the kitchen of the cell. Plastids. A very essential thing to remember that plastids are found only in the plant cells, not in the animal cells. Another important aspect that they also contain double membrane bound covering. So there will be one outer membrane, there will be another inner membrane. But the inner membrane is not folded like in case of mitochondria. But plastids themselves are very specialized structures. So before I go into the detail of this structure, which is the chloroplast, let us talk about the plastids in general. So plastids are present only in plant cells, as I have just said, and there are two broad categories: chromoplasts, the colorful ones, and the leucoplast or the colorless plastids. Now chromoplasts, according to the pigment that they contain, can be further categorized into two categories. Those which contain chlorophyll pigment, which is a green pigment, they will be known as chloroplasts. And chloroplasts are important for photosynthesis in plants. So photosynthesis is that process which results in all the biomass formation, which results in the formation of oxygen that we breathe, and that's how chloroplasts are very important plastids. Now, along with the chlorophyll, chloroplast will also contain some yellow or orange pigments in addition to the chlorophyll. But since chlorophyll amount is much more than these pigments, that's why it appears green. Now let us talk about chromoplasts. Chromoplasts are those plastids other than chloroplast which contains yellow, orange or red pigment. Now these plastids which contain colorful pigments are helpful in giving color to the flower and fruits, and in flower they help in bringing or attracting the insect for pollination. In fruits they attract various animals which help in the dispersal of seeds. Then, what is the function of leucoplast? As it doesn't contain any pigment, so it contains food materials for storage. So, it might have starch, oil and protein granules stored within them, and accordingly, the starch containing leucoplasts are known as amyloplasts, oil containing ones known as elaioplasts, and protein containing leucoplasts are known as eleuroplasts. So plastids are only found in plant cells. They can be containing pigment or the stored food. The pigment containing ones, if they have colorful pigments other than green, then they help in the attraction of pollinators. If they contain the green pigment, they are known as chloroplasts, which are involved in the photosynthesis. Now let us learn a bit more about the chloroplast structure. So as I just told you, plastids are double membrane bound structures, and in case of chloroplast, inside this membrane, whatever the fluid is filled, it is known as stroma. So stroma is not an ordinary fluid. Also, what else do we find here? That stack like arrangement of various membranes. Now each stack is known as grana, and the membrane forming these stacks are known as thylakoid membrane. Now you can also see that one grana is connected to another grana. So this connecting membrane is known as stroma thylakoid membrane. And this membranous system along with the stroma completes the entire process of photosynthesis in plants. So remember, we will discuss the details of photosynthesis in 10th standard, but it completes in two major steps: light reaction and dark reaction. And light reaction takes place in this membranous system, while dark reaction takes place in stroma. So chloroplast is a very important kind of plastid which has a certain kind of membranous arrangement inside its both the membranes. Now, remember that just like mitochondria, plastids are also not only double membrane bound, they also contain their own DNA and ribosomes, which makes them autonomous. Now let us go to vacuoles. What are vacuoles? So, they are storage sacs for solid or liquid contents. Sacs and for storage. Simple. Now, where do we find them? So, we find them in both plant cells and animal cells, but in plant cells, vacuoles are very large, and remember, they might be covering almost 50 to 90% of the total cell volume, depending upon the stage of cell. And in plants, what do they contain? They contain cell sap. So this cell sap will provide turgidity and rigidity to the cell. They also contain amino acids, sugars, various kinds of organic acids, and some proteins, and they store them. Now another function of vacuole is food storage, osmoregulation as food vacuole, and excretory vacuole. These are the various types of vacuoles, and these functions mostly pertain to the animal vacuoles. Let us see how. So, animal cell vacuoles will be smaller in size, and they will not be permanent as in case of plant cells. They will appear and disappear according to their requirement by the cell. And what are those functions? For example, simple single-celled organisms like amoeba engulf their food item, and that food item engulfed inside becomes a food vacuole. Now that food vacuole is fused with the lysosome, the food is digested and absorbed, after which that vacuole becomes an excretory vacuole. Also, in some other unicellular organisms like paramecium, etc., specialized vacuoles are also present which help in removing the excess water from the cell, and they are known as contractile vacuoles. And some vacuoles also help in waste disposal from the cell. So, vacuoles are basically sacs. Now, depending upon plant or animal cell, their size and their functions will be very different. But remember, in plants, they will be very big, pushing all other cell organelles towards side, and they will be permanent. In animal cells, vacuoles might be temporary, smaller, and their functions will be different as compared to the plant cell. With this, children, we are stopping our lecture now, and in the next video, we will be talking about some other cell organelles like peroxisomes, ribosomes, centrosomes, and a major topic, that is cell division. So please revise all this and come prepared in the next lecture.